because I think everybody interacts with the stress axis. We typically think of stress as negative, but remember, right, this is really a challenge to the system. So those challenges, right, I think of the way that you get better at things is to expose yourself to challenges and overcome those challenges, right? So being challenged, right, having the system be challenged is good for the system in many physiological variables, right? So for example, I mean, you could even think of the simplest form of this. People who exercise live longer than people that don't, right? Notice that exercise is a stressor, right? And repeated exercise leads to better vitality and a longer life, right? So stress versus not stress at all, right, is actually a net negative for the non-stressed individual. However, excessive stress leads to increases in all-cause mortality, right? Cardiovascular disease, metabolic disease, and all things are sort of downstream of that. So we can think about acute versus chronic, and we can also kind of look at this from the sense that if we think about the commonality of the stress response, notice that the first thing that any problem needs is the resources to solve it, right? I mean, I think that's really the kind of essential logic of the stress response's similarity. For almost all problems, you can be assured that you need resources to solve that problem. Now, we can think of this at sort of like at an individual level. If some problem arises in your life, the magnitude of that problem, like how big of a deal it is, depends often on the amount of resources that you have available to devote to it. If you're already stretched thin, any small problem on top of that can become a very large problem. But if you have plenty of resources, whether that be money or time, to address a problem, you can easily deal with it, right? So this is a kind of, we have to throw resources, and so both alarm phase and resistance is all about generating the resource currency of physical systems, which is energy, right? We're going to throw energy at the problem. Whatever it takes to deal with, we know that it's going to require more energy. So typically that involves tapping our reserves of energy. Okay, so typically we think of stress as being a net negative for the immune system. But the reality is that's not always the case. And so we see a kind of bifurcation here with the stress response acutely. Notice that acute stress increases several measures of immune function. Now this is a percentage, right? So the way that you would read this is these individuals are being exposed to an antigen, right? So in experiments like these are typically sort of like dead bacteria, things that are not themselves infectious, but generate an immune response. And so the way these experiments are done is that you induce a stressful experience, right? And then you expose to an antigen, and then you measure the immune response to that antigen over a series of days, okay? Now, what we're looking at is an intact, and particularly important for our stress research is that we do the sham as well, right? Because unlike a lot of these, what we're here, we can see, is there an effect of sham surgery? Yeah, there is, right? So the surgical procedure of an adrenalectomy is itself stressful. I don't think that should surprise anyone. But what we can distinguish between is the added effect of the adrenalectomy itself. Now, so notice, what hormones are we taking away with an adrenalectomy? Cortisol and epinephrine and norepinephrine. So we're taking away, good, we got them all, right? So we're taking away pretty much all of the hormonal response of the stress axis, right? And what we can see is that if we expose individuals to the same short-term stressor, we do not get the stress-induced increase. Okay? So, in an intact animal, if you give the animal a moderate stressor followed by an antigen, notice that you have an increase in the immune function that is rather significant, right? So we go from essentially a 3% change in immune function. I mean, these are, right, there's a number of measures that these could be, like T cell counts and all this kind of stuff. So you can measure various attributes of immune function. So we go from a 3% increase or response essentially to the antigen compared to a 5% increase, right? That's like what, 40% more or something like that? Now, the sham knocks down both of those responses. Right? And so what kind of stressor is surgery? Would you say it's acute or chronic?
Acute.
I would say you could, yes, so there, I can see why everybody says acute, but notice that from a surgery, the challenge is the recovery period, right? So acute stressors are typically going to be measured in hours at most, right? So while I would say, yes, going in for surgery is both maybe an acute and a chronic stress, but I want to emphasize that it's typically the chronic stressors that negatively affect the immune system. And so when we can see this lowered response in both cases, we're really looking at the effect of chronic stress. And we're trying to really overlay the effect of acute stress on top of that. And we still see this effect, right? We still see the effect of a brief acute stressor prior to the antigen administration, producing an increased response from our immune system. That is completely abolished by adrenalectomy, right? So, and again, remember, we're not measuring, right? The graph is not depicting, right, like cortisol or any hormone. It's actually depicting the immune system's response to the antigen. And so in the absence of the stress hormones, notice that we don't see the increase induced by short-term stress, okay? So the takeaway here is that brief sort of acute stress can actually increase immune function through the hormones of the adrenal gland, right? We can't say which hormone it is, but we can say, and again, so if we release epinephrine, do we necessarily release cortisol as well? Yes, that one's true, right? If you release epinephrine, you for sure release cortisol, right? You can't get away from that. But you can have cortisol without epinephrine, right? I know that's a tricky one, but epinephrine is always the fast response. The fast always includes a little bit of the slow, but you can have a gradual increase in cortisol secretion all by itself. Yes?
Okay, I kind of missed the takeaway. What was that sentence that you wanted to take away?
That acute stress can increase immune function, right? So and the reason I think this is important is because typically, we think of stress as being negative for immunity, but this shows that within certain contexts, right? Not chronic stress is still a net negative, but acute stress, right? Okay, so we're gonna talk a little bit about what we call developmental stress. And so developmental stress is kind of an umbrella term. It can refer to like the sort of early life period, right? So if you're looking at stress when you're a juvenile, that would be considered developmental stress. You'll also see the term used to describe things like prenatal stress, right, as being developmental stress. And so again, I think sometimes it may be better to specify whether we're talking about in utero stress or not, and certainly each individual experiment would. We can sort of generally say developmental stress is either happening in the womb or in the early life period. Okay? And so one of the sort of very, I'd say, I don't know if you'd say it's famous or infamous, are Harlow's monkeys, right? These are the ones where the babies were removed from their mother and raised by essentially wire or plush replacements. And you can see from this picture, the picture on the left, and you might not be able to tell this, but the picture on the left, the sort of chicken wiry cylinder, has a nipple protruding out of the center, which contains food, right? Now that's a kind of a critical idea here, but notice that the monkey is actually on the other one that has the soft towel wrapped in I'd say a more monkeyish face on the top. And so the surprising finding was that the babies preferred the plush-ish, I wouldn't even call it necessarily plush, because it's really just a towel wrapped around chicken wire. But I would say that what was interesting was that the monkeys preferred the pseudo-social connection even to food, right? And when those monkeys grew up without the influence of any kind of social development, you can imagine that they were both socially inept and also overly fearful and anxious in social environments, right? So this kind of spawned a field, and again, no one will ever be able to repeat this again at any major, Why are you looking?
Oh no, I was just wondering why.
Why?
Yeah.
Because this
monkey...
is essentially a form of torture.
Right.
Right? This would never pass, right? Any kind of animal care and use, because so you can do pretty much, as a general rule, you can do pretty much to any animal, do anything to any animal that doesn't have a backbone. Like, you could do this in fruit flies, but fruit flies don't care, right? Fruit flies don't have any parental care, right? But as soon as an animal is a vertebrate, you are limited into what you can do with it. And when it gets to primates, you are severely restricted for good reason, right? So these are very sensitive, cognitively sophisticated animals with like an internal life of their own, and you're essentially torturing them during their developmental period and asking these questions. So I think these experiments done once are sort of enough, right? We know, at least in part through this, that there is a significant impact on the social development, the brain development. Yes?
I'm kind of missing the torture part. Are they taken away from their parents?
Yes.
Okay.
Those things that you see there are their parents.
Okay.
Right? So the chicken wire mom and the towel mom, right? That's the only interaction that these animals have, right? Or did have.
Yeah.
With this famous experiment, I thought when I heard about it when I was younger that it was significant because even though they had like nourishment, because they didn't have social interaction, that they end up actually dying, even though they had.
They don't die, right?
No, I thought I remember.
Yeah. So the problem is that so monkeys, much like us, are very socially active, right? And so one of the stunning findings is actually the one that's picked here, right? Which is the animal will essentially starve, right? And instead of going for the chicken wire mother, right? They prioritize any kind of social contact.
Right.
And so if you think about, you remember the discussion we had around skin-to-skin contact for primates, right, including humans, that how important that is. And also when we looked at parental care in primates, notice that all the primates do is, the only thing that they do is cling to mom. That's all, right? And obviously there's nursing and things of that sort, but their mindset appears to be just to cling to mom. Now, I don't want to go into this experiment in great detail, but I just I'm using it as an example to show that these things have rather significant consequences. So let's talk about this one more instead, right? Because this is actually an experiment that we could run and we could continue to do. And this has a really kind of critical message here. And I like these studies, which I think that allow us to sort of a window on human behavior and policy, right? Because we often lose sight of the fact that we are biological creatures, right? And so this is an interesting one. This is about prenatal stress and the effect of prenatal stress on the lifelong stress reactivity of the offspring, okay? Now I know that was a mouthful, but basically the idea is what we're interested in finding out is whether stress in the womb, right? So this is developmental stress in utero, affects the adult in significant ways, okay? And so what we're interested in is adult stress reactivity. And probably the best way to think about stress reactivity is that each individual responds to stressors differently. So one individual might, for example, like roller coasters, right? And another individual might be terrified of roller coasters, right? So individuals might be scared of heights, right? Or not. But to a stressor, the response, right? If you give, let's say, 5 people the exact same stressor, you're going to get 5 different responses. Now, the interesting thing is that within an individual is usually, we could say, they're either highly stress reactive, like really jumpy, or not, right? And so those things tend to be consistent over the course of your life, right? So whether you're highly stress reactive or you're low stress reactive, that tends to be a stable trait, okay? So it's like if you're jumpy as a kid, you're probably going to be jumpy as an adult. If you're not easily spooked as a child, you're going to be not easily spooked as an adult. And so what we're asking in this question, and this is done in rats, is does prenatal stress affect the stress reactivity? And we're going to measure stress reactivity through just measuring corticosterone, which is, right, so that's the rat version of cortisol, right? It's a glucocorticoid, it's their primary glucocorticoid. So what we're going to do is give the rat mothers who are pregnant at this time a restraint stress, which is not that, like, it certainly doesn't hurt the animals at all. It basically, you put a rat in a tube and you prevent them from moving. Right? This is very stressful to rats. Right, I imagine we would not like to be plucked out of our seats and stuck in like a straitjacket for some amount of time. I imagine that would induce a stress response in us as well. We're going to do that, and we're also going to take one step further and basically do the same thing we saw before, which is adrenalectomize. And again, why are we going to adrenalectomize these animals and run the same experiment? To see if it's the hormones of the adrenal gland, right? Which is, it's going to be hard for it not to be, but that doesn't mean that we don't have to verify that it is, right? Okay, so if we're interested in whether or not prenatal stress affects adult stress reactivity, right? What is the critical comparison here? What's the first critical comparison?
I was literally going to ask the same thing. Yeah.
Okay, that's why we can pause and we can go again here, right? So if we're interested in understanding whether or not maternal stress affects adult stress reactivity, what is the comparison that will answer that question? Yes?
Okay, are you generalized to the adrenal intact?
Not quite. Not yet. Okay, so again, remember, the first question we're asking, question number 1, is does maternal stress affect adult stress reactivity?
Okay.
Is stress versus non-stress?
Stress versus non-stress in adrenal intact animals.
I didn't
Okay.
understand what's that. Okay.
All right? Now, because notice our question is kind of in parts, right? First off, does maternal stress affect offspring, right? Stress reactivity. So in order to answer that question, we need a comparison between an individual or a group of individuals who was stressed, or whose moms were stressed, and an individuals whose moms were not, right? Does that make sense? And so, does maternal stress have an effect on stress reactivity in an adult?
Yes.
What is the magnitude of that effect?
Twofold.
It's about 2.5x, right? So again, I keep trying to emphasize that we don't say stress has an effect, right? We say stress, right? Prenatal stress more or less triples the stress reactivity of the adults. Now, to get to the second part of this question, is that response mediated by the hormones of the adrenal gland?
Yes.
How do we know? What are we comparing here?
Intact versus adrenal ectomy.
Yes, right? And adrenal ectomized versus adrenal intact, right? So we know this is what should happen if the adrenal glands are intact, and this is what happens when they're not, right? Does that make sense?
It does.
And so there we can say confidently that maternal stress affects adult behavior, and that effect is mediated by a product of the adrenal glands. Right? Does that make sense? And we can point to the data which suggests that that is the case. Now, why might this be important? When is one of the most important times? If we want to think of as a society, we think stress reactive people are generally going to be less social, right? More like antisocial. When is probably the most important time to intervene in a person's life?
Pre-puberty.
Probably in utero.
Okay.
Right? Now I know that's kind of a controversial take, but there's data to suggest that some of the biggest effects that we've seen are happening prenatally. Right? So if you think stress reactivity is a net negative, the important interventions happen before that individual is born.
So this is done, I'm assuming, to help mothers have more successful pregnancies. Like, what is the government, or who says, okay, we see this is going on, let's intervene?
Well, that's unfortunately that ultimately is left to politicians whether they should or shouldn't intervene. But again, I think the larger point is when we talk about societal issues that interact with biology, we should always be able to articulate a position that's based on biology, right? So this is in rats, right? It's not in humans. But what it shows is that one of the things that has the biggest effect is the maternal stress of the mother. This changes, right? It changes stress reactivity, it changes many aspects of physiology, right? And so if we are deciding between, let's say, like, oh, maybe we should fund pre-K, or maybe we should fund, right, maternal care ahead of time. Someone might say, oh, I think it's gonna have a bigger impact on these kids if we go pre-K, and you could, and not that you would say this, but you might say, someone looking at this might say, well, actually, I think we could have a much bigger impact on the sort of lifetime outcomes of the individual if we make sure mom is taken care of, right? I think that's kind of generally the case, right? So maternal, like in utero nutrition is one of like the most important things that you can have, right? Stress levels for mothers is one of the biggest impact things that you can have. And you can point to data like this. Now it's not in humans, right? But there's no reason to expect that this would not be true in humans. You had your hand up, right?
Yeah, I was wondering why the non-stressed mother in the minus one is higher than
Well, so here's the thing. In both of those cases, mom has been adrenalectomized. And whenever you're at real animals, and one of the things that I don't love about the way the book presents figures, if there's one criticism I would have, it's that they never put error bars on any of their main figures, right? So it's highly, like looking at that's not a huge, I mean, we're looking at maybe 3.5 to 4.1 or 2. I'd say that's noise, right? But notice so we're gonna take the adrenals out before pregnancy, so there might be some effect of that, but I'd say that looks more like noise to me, as does the difference between non-stressed and stressed in the adrenal ectomize. That's probably noise or very close to it. I don't think that, so notice that if that effect was real, right, like actual of biological significance, what it would mean is that stressing a mother who doesn't have adrenal glands decreases the stress reactivity of the offspring, right? That's a tough set of things that would require a lot more experiments. But if we thought that that was what was actually happening, if that was consistent over multiple experiments, maybe we would go and at least think about what in the world could be causing this. Which doesn't mean that there isn't something, it just means that when we look at this data, we have to kind of evaluate that on a case-by-case basis. But I don't think there's any, I think that's noise, right? Yes?
The plasma corticotestosterone, is that the stress response in the milk? Is that what's measuring?
Yeah, it's just a glucocorticoid, right? So it's equivalent to cortisol. So our stress hormone, our main stress hormone is cortisol, right? But rodents, they don't release cortisol as their main stress hormone. They produce corticosterone. It functions essentially identically, and most of the time, to be fair, I just say cortisol, regardless. Much like you said estradiol or estrogen, we just call it estrogen, right? Because there are species variants, but this is their stress response, right? And these are tested as adults, not as juveniles. Okay, another example of what we would throw under the umbrella of developmental stress is a really extreme phenomenon, which is called psychosocial dwarfism. Now, talk about torture, this is as close to Harlow's monkeys, I would say, as you're going to get in humans. So these were, this woman was, she was at essentially an orphanage in Eastern Europe and basically got no physical contact with anybody for most of her life. Okay? So, and now again, we can, and I think we would all agree that's a tragedy and it's horrible and all of these things, but what we can look at is, first off, how this affected her development. So the one on the far left, right? So this is 15 years old, right? And the far right is about a year after this, right? A little bit over a year after, maybe a year and a half later. So that is a lot of development in a single year. So what is going on, and does this relate to developmental stress? So what we're going to be measuring here, rather than glucocorticoids specifically, right, is we're going to measure growth hormone, growth rate, and we're going to qualify that, right, by measuring caloric intake, right? Now, why is caloric intake an important control here?
Gotta have macronutrients.
Yep. Yeah, so, but why do we need to control for that?
Because there must be like a return.
Yeah, so another way to think of this is, this person who we just took from one of the worst conditions you can keep a human being in might not have been getting enough food.
Right.
So if they suddenly go on a huge growth spurt, we could say, well, how do you know it wasn't that they just had food that they could eat now at this time, right? So this is why we are controlling, we're controlling for food intake, right? Because what we're gonna see, you can tell, she is a lot taller, right? And so if we look at growth hormone concentration, right? 5.9 nanograms per mil, right? And the growth rate, so this is centimeters per 20 days, and you have your caloric intake, 1,600 calories. Okay? So, is she starving? No, right? So that's important because that means that the effect, the physiological effect, is not a nutritional deficiency, it's not a lack of calories, right? Because if you starve a child, will they grow more slowly and develop slower?
Yeah.
Yes. Okay, so as long as we're on the, and this is not what's happening here. What we're talking about here is how a psychosocial or a psychological effect is dampening down, right, development. So, 100 days after admission, notice that the calorie count is actually down and the growth hormone and growth rate are up, right? So we've doubled plasma growth hormone and tripled our growth rate. Now, I feel like after the life that this woman must have led, to have your favorite nurse go on vacation, you know, and I feel like also that it must have been for a pretty long time, right? Because we're actually, so is the removal of her favorite nurse a stressor? Can we see that? Yeah, it's a massive stressor, right? And notice that we're essentially dropping it back down again to essentially admission levels. But when the nurse returns, we get, again, the same rebound effect. So what I want to point out here is that this is a psychological stress effect, right? If we think about growth and development, again, normal growth and development, we think of, oh, okay, or is this individual too small for their size? Well, are they eating? What are they eating? What is their nutrition, right? Why are they not developing at the same pace? But we can see from this that there is a psychological component which can severely retard growth. Okay, everybody clear on that? I think it's a pretty dramatic example. Now, we go back here, this is some of the work, yes?
This is making me think. I don't really know how to like pose it in a appropriate question, but like if you look at like NBA players and stuff like that, like they'll talk about like, oh, this one summer I was, 6'1", then I was 6'9" that summer. See what I'm saying? Like, how does that relate to the conversation?
So that is probably just the growth spurt that individuals hit, right? So
how could it not be something that she's experiencing? Like, oh, if
because
they'll do it
so
in the summer,
difference is,
okay,
so
boom.
typically, so at least what would cause us to suspect that something was going on, right? Maybe a better example than an NBA player hitting a huge growth spurt, right? Is like there's two forms of dwarfism, right? There is a kind of genetic dwarfism, and then there's an endocrine dwarfism. Right. So the endocrine dwarfism is caused by low levels of growth hormone. Right? Then it's measurable, right? So earlier you said you had kids, right? So when you take them to the pediatrician, they get, just every time you go in, they measure how tall they are, right? And what they're trying to do is measure like what quartile of growth they're in for their age. And so if you are in the lower group, you're going to get tested for growth hormone, right?
Right.
Now, this effect for psychosocial dwarfism is an active inhibition. So our 15-year-old girl would be in that bottom quartile. Now, right? So if you think of dwarfism, right? She has dwarfism at that particular time in her life, but it is caused by a social phenomenon, right? It is not the same as an individual hitting a growth spurt, being essentially normal, and then turning on the HPG axis, right? And then hitting a massive growth spurt, right? That is very different, right? Because the difference is that it's the difference between a positive stimulus, right? So testosterone, IGF-1, right? You're gonna grow. Like, if you give any child IGF-1, they will start growing, right? But the difference is those individuals did not start out inhibited.
So I have this person. She's 4'11". She's an adult. So, if she's short, they're gonna test her for growth hormone. Would she have low growth hormone because she's short?
Well, some people are just short.
Yeah, that's what I'm saying. Like,
so that no.
But she would
so-
have regular
so-
levels
so
of growth hormone,
essentially,
right?
so there's two. So here's the thing. There's really two components to growth, right? And I don't want to divest into this too far, but there's two components to growth. There's a genetic height, like you're quite tall, right? Other people are not as tall as that,
Yeah.
and they will never be as tall as that unless you were doing a kind of thing like Andre the Giant, right? Everyone knows Andre the Giant. He had a pituitary tumor which produced too much growth hormone and too much IGF-1, which is what caused the gigantism. But so he exceeded what his genetic height would have been, right? So I'm like 6 feet tall, right? So if you treated me with growth hormone as a child, maybe I'm going to exceed that a little bit, but if I have normal amount, you're going to be your genetic height, right? So the case of 4'11", I think the cutoff for dwarfism is like 4'9" anyway, because
So-
at that point you can get a handicap space.
so do short people? Do they have?
I know that because I had a girlfriend who was 4'10". Right?
So-
So I was like, can I get a handicap space? I was like, we looked it up. Apparently the answer to that is no.
So-
This is the cutoff.
So short people that are not dwarfs, they have normal.
Yes,
pH
normal
level.
growth hormone. They just have a
Okay.
genetic cap.
Okay, thank you.
Okay? All right. Let's get to some of the brilliant work of Alison Fleming. So this is one of the kind of best stories, and we've kind of encountered this already. This is the a kind of revisiting the high and low licking moms, right? So if we remember, we have high and low licking moms, and so this is the epigenetic transmission of behavior. So a high licking mom will have high licking daughters, right? And a low licking mom will have low licking daughters. It is not genetic because we can cross foster, and whatever the behavior that you expressed, right, or you received as an offspring tells you what kind of mom you will be. So we looked at how that leads, if we remember, to the methylation of the estrogen receptor, so that if we kind of start piecing it all together, remember estrogen in the POA facilitates parental behavior, so the less parental moms, right, the low licking moms, don't have as many estrogen receptors in the POA, right? So that's again, that story was, it's pretty compelling and remarkable. But there are other effects of this as well, right? So high and low licking moms have offspring with different stress reactivities, right? So different stress reactivities. And so if we remember our concept of stress reactivity, we just talked about it. Notice that what we're measuring here is corticosterone, and this is ACTH. It's corticosterone, just as a reminder, because this is the glucocorticoid in rodents, right? That we see low-licking moms, the offspring of low-licking moms, have higher stress reactivity than the offspring of high-licking moms.
Say that again. The bottom one is what? Cordo what? I can't see it.
Corticosterone. So you should think rat cortisol,
Okay.
right? It's the same, and again, so our primary glucocorticoid, which is our primary stress hormone, is cortisol, but for rodents, it's corticosterone. So those are more or less equivalent. Okay? So we understand what's going on here?
Okay,
Yeah.
you said the offspring of the low licking moms
Mhm.
have higher,
Well, I didn't really say that.
okay,
They said that.
have higher responses with the high licking, right?
Well, let's look at the data, actually. Why don't we, I love the intro, why don't we look at the data? So what are we, so if we could reformulate what you said as a question, right, we could say, do high-linking moms have low stress-reactive kids? And conversely, low-linking moms have high stress-reactive kids? Right, those are question marks,
Yes.
right? So, how do we know?
Based on the data, the low maternal looking offspring had higher levels of.
How high, how much higher?
Like 200.
What,
Yeah, so I'd say, you know, if we think about how much higher is that, right, after the stress, let's take the post-stress period, how much higher is it? It's about 45 to 60, that's about, what, 15, right, 15-ish, right? So 15, that's about, what, 30-ish%, around 30% more, right? So again, notice that to formulate your question, this is what we should be doing, right? And again, the whole point here is that while it's true that I am saying them, I didn't do these experiments, and the answer is in the data, right? We should always think that way. The answer is in the data, not with what anybody tells you. Because the more papers you read, the more you realize sometimes they'll say something that is not in the data at all, right? So, what we can see, and we'll just look at corticosterone. So we say it's got about almost a 30% increase in corticosterone after the same stressor, right? So, and again, so high licking moms have low stress reactive kids according to this, right? So, did we answer the question sufficiently? Yeah. Yes.
So, is it environmental because, like you said, you can change out the mom and
Is it environmental? You tell me.
it is
So what's the other option? Is it environmental or is it, okay, so what is it?
Or what? Environmental or what?
Genetic, innate, right? We don't even have to use genetic. We could say, is it innate or is it environmental? So, which is it? It's environmental, right? Again, this is, and to kind of link it to the umbrella we were talking about, developmental stress, right? So this is an example of how development, right, i.e., and again, this isn't taken like, it's not like the pups are being licked and then we go and stress them and measure it, right? These are the pups that experience something early in life, we let them grow up, and then we give them a restraint stress, and we ask how strongly do they respond to it. Yeah, see, it says genetic right there. So it's definitely environmental, right? Now, we have our old friend, and we're not gonna go through this in great detail, but it's roughly the same mechanism. So epigenetics has been a sort of boon to the field. Notice that with epigenetics, we have unique epigenomes that are typically responsive to the environment. So these represent gene by environment interactions. And so what we are seeing is that in the high licking pups, the adult, right, has lots of glucocorticoid receptors in the hippocampus and low glucocorticoid levels, right? And so we can actually trace this back to a decrease in the methylation of the glucocorticoid receptor, right? So notice we went from behavior to hormones to epigenetics to explain a phenomenon that involves, again, both estrogen and glucocorticoids, involves parental behavior and stress reactivity. Okay, so stress, I think, I'd like to have hoped to have convinced you at this point that stress can be a positive, right? And that if There is, however, kind of an optimal range for stress being positive. Too little stress, you're not generating an adaptation. Too much stress, you're actually decreasing many of the factors that you might want to increase, right? But certainly stress can be a positive. This is kind of another way to kind of think about it. You can have acute stress, right? Most of the time, again, as long as it is not too severe, right? Because obviously you can have a massively severe acute stress that it leads to long-term problems. But again, as long as it's not too severe, right? You can get enhancement of immune function, enhancement of androgen secretion, enhancement of growth hormone secretion. But as we move from days to months, everything gets negative, right? Days may be okay, but as we move towards, I should say, weeks and months, then we are getting into that sort of range of chronic stress, whereas months to years, really there's really nothing good there from being stressed for a year, right? There's no positive, except that you're still alive. Okay, so we have a couple other things to talk about. One of them is social rank, right? Stress and social rank. Now, before now we reading the slide, is it more stressful to be alpha or beta, or what's the least stressful position in a hierarchy?
Okay.
We got beta, maybe.
Alpha.
Alpha, maybe.
Oh, I guess they both come with their own stress.
Well, I think what we'll see is that it depends, right? It depends a lot on, and this is really the stress level of everybody in the hierarchy, right? If it can be the least stressful or the most stressful to be the alpha, right? If you're in a very stable hierarchy and everyone's submitting to you, then chances are it's not that stressful to be the alpha. But if you're constantly looking over your shoulder, right, the sort of heavy is the head that wears the crown kind of thing, right? If you're constantly worried about being overthrown, then it can be the most stressful. And so depending on what species you are looking at, right? So A is the African wild dogs, right? So these are, this is corticosterone again. And so dominant and subordinate individuals, at least for males, don't have a massive difference, but being the dominant female is very, very stressful. Who runs the pack? Let's look at the data. What do you predict? Who's running the pack?
Female.
It's the females. Are they comfortable in their position?
No.
No. Right? It's very stressful to be the dominant female, but you know, we are going to see some differences so this is in rats. And what we're looking at is, this is male rats. So who's dominant in a rat group? Is it males or females? It's males, right?
What?
Males. So again, so for African wild dogs and hyenas, it's females, right? And again, this is, what's interesting about these is we would typically call those like sex-reversed species, but you know, hyenas are the really kind of famous example of this, right? Where the lowest ranking female has higher status than the highest ranked male, right? Females are much more aggressive, right? These kind of things, you know, so matrilineal society, it's not like the females are not aggressive, they're actually typically more aggressive than males. But when you see something like this, you can be pretty confident male rats are bigger than female rats, right? They are more aggressive, we've seen that a bunch. Is it more stressful to be dominant or subordinate if you're a rat?
Subordinate.
It is more stressful to be a subordinate. And notice also that the dominants here have higher levels of testosterone. So, and the sort of kind of point here is that there is not a clear, so it's not always dominant is the most stressed. It's not always subordinates are the most stressed. You can have even situations where stress levels don't correlate to social rank at all. Right? So, why might it be more stressful to be a subordinate? What? Yes.
You work harder for protection and normal problems that you have competition.
Yeah, so that's, yes, it was over, someone said
I was
something.
gonna include like the reproductive, like in the success rate, maybe.
Maybe that's, so one thing that's always important to remember is that we worry, they typically don't. Right? Worrying is a very primate-y thing to do. Right? Yes. Well, I think it's not even that they don't need to imagine that or think about it. They're getting harassed all the time.
Right.
Right? So yes, that's the kind of the point is that when you see animals have something like this, it means that that animal is experiencing aggression a lot. Right? They're getting bitten, they're getting excluded from food, right? And so we would worry about it, I think, if we were them, but they are, maybe it's a gift that they can only experience like what's going on right then. You know, you've got a kind of an integral function maybe that's kind of, that you could look at as as a proxy for what their life is actually like. But one of the kind of major things about hierarchies, in addition to the amount of interaction between individuals, is how stable that hierarchy is. Right? So hierarchies that are stable tend to have lower cortisol levels for everyone. Right? And now again, there are exceptions, right? If you're being harassed all the time, it's terrible to be on the bottom of a hierarchy. But most hierarchies are not like that. Now, if we think about the way hierarchies typically work, who's doing most of the harassing?
The challengers, the non-dominant.
So if we think about hierarchies, right? So imagine how, like if you have a multi-level hierarchy, how often are you getting a direct interaction with the alpha?
Well, you're competing for beta, yeah. They always like, oh
Well,
no,
for
I'm trying to
beta
win.
is one level down.
Yeah.
So, but typically if you're like on 3 or 4 rungs down, is the alpha really even?
Oh no,
No,
no issue.
right? Who's harassing you?
The people in
The
proximity.
immediate individuals above you.
Hmm.
Right? Now, what we talked about a little bit about this hierarchy stability, and the whole reason that hierarchies form is to essentially reduce conflict, right? So aggression is conflict resolution, right? So we reduce aggression by reducing conflict. Okay, so why is it not in the best interests of, let's say, the level right above the lowest level to constantly harass the lowest level?
They don't have anything.
Well, harassment is generally not over something.
Because the lower level don't have anything to harass.
The lower levels aren't going to harass, but what happens if, let's say, right, you, like I'm the lowest level, you're 1 rung above me, and there's like 5 rungs above that. What happens if you harass me to the point where, like, I just shut down and leave the group?
They're the lowest.
Then you're the lowest. Have fun with that. But that's the idea. So notice that there's an incentive built in most cases, to not harass to death the lowest people, right? Not people's the wrong word. the lowest individuals. Now, the reason for that is, and this is why, in most cases, the benefits of being the lowest member of the hierarchy are better than being on your own. And the lower rungs of the hierarchy are incentivized to keep it that way. Right? Because if you, right, notice if you're, the second to last member of the hierarchy and you harass to death the person below you, then now you're the lowest member of the hierarchy, and that's not a great position, right? Yes.
But why would that matter? Like, you still under everybody you was already under, so what's the difference?
So that's not necessarily true. Now, first off, keep in mind that we all tend to do, you're anthropomorphizing this. Right? So all I'm saying is that at every level of the hierarchy, you are incentivized most of the time to not be overly aggressive to your subordinates. Because there's two factors. Maybe in the future, those relationships will change. And third, right, you do not want to be the lowest person if you are already above on the hierarchy. Okay?
Yeah.
Right? Animals are always trying to climb in rank,
Yeah.
and that is essentially a demotion.
Gotcha.
Okay, I want to go through a couple more slides here quickly. So the model for humans is essentially the baboons, and the issue here, this is a kind of well studied by again, one of those famous behavioral endocrinologists out there, Robert Sapolsky. He studies baboons in their native environment. The thing about baboons is that food is abundant and nothing eats them. So really the only stressors are other baboons, right? And they harass each other all the time. And so the dominant males have high levels of testosterone, and when they have cortisol spikes, and this is a typical pattern that you see in dominance in humans and all kinds of animals, is that cortisol levels spike and come down quickly, whereas in subordinate individuals, cortisol comes up slower and stays on longer, right? So from the alarm phase, if you are having the best possible stress response, high cortisol, and then you drop that cortisol quickly. And again, for subordinates, higher baseline cortisol, higher, again, slower, longer for glucocorticoid response, high blood pressure, low HDL, right? Suppressed immune system, low growth factors. So this is what chronic social stress does to baboons, and really is what chronic social stress does to humans. And this is why this is a valuable model for us to kind of look at this. Okay, a few points before we get into maybe we can do one more figure. Basically, short stress is good, long stress is bad, right? We've talked about social isolation before, so we said solitary confinement is about the worst punishment that you can do to any social animal, but notice that many times the elderly become socially isolated, right? We talked about in social behavior, we talked about dogs and these types of things that can positively affect social interaction. Let's see, a couple, you can read this. I want to do this study and then we'll kind of go into the review session, because this is a really kind of critical idea. And there's kind of one other big study that I want to talk about, but this one is a really important one. So, I want to set this up. These are wives with husbands. If they're the caregiver, the husband is terminally ill. Okay? So the blue, the yellow line's not supposed to be there. So just keep that. The yellow line means nothing, right? However, right, the blue is just age match controls, and the red are women who are caring for a terminally ill husband. What they're going to do is they're going to take a punch on the arm, right? They're going to generate a wound of equal size on everyone, okay? Right? Now, what we are going to be measuring is the percentage of subjects with completely healed wounds over time, okay? So, if we, just so we're reading this correctly, by 4 weeks, how many control women had their wounds completely healed in a month?
20%.
Yeah, about 20, so about 20%. Now, notice that this, for both groups, has to get bigger over time, because everyone that is in here is also here, plus all of the new people that were healed. Right? That make sense? Okay. So, is there an effect on the immune system of caring for a terminally ill spouse?
Yes.
Yeah.
Yes, right? Now, where is that most noticeable?
7.
Really the first 6 weeks, right? So in normal controls, right, this is age-matched women, right? 50% of them are completely healed by 6 weeks, whereas how many of our caregivers are healed by 6 weeks? Like 15%, that's a huge difference. And so the other kind of piece, and we can, look at week 7 and 8, but notice that by week 8, that's 2 months for a small skin lesion. I mean, this is not a massive wound. That you're only at about 85% of the caregivers, right? They still have not healed when 100% of the normal women have healed, right? So stress can, and this is kind of one of the most stressful human situations I think that we can imagine. If you've ever cared for somebody who's terminally ill, it is very, very stressful. Okay, so I'm gonna stop there because I have your exams to give back, and we can talk about them, or we can do questions as well. Right? Yes.
So, when you're formatting this in a takeaway, I don't want to say it does have an effect.
I went to his office.
Right, that's good. That's right. You don't want to say that.
However, is what I say
I didn't look. I
stress
refused.
can,
I was scared.
inhibit wound healing, or I kind of
I would say, decrease, right? So that's about a 3 times effect, right? As I keep stressing to you guys, and this is something that I emphasize it because I hope that you take it with you past this class. Whenever you see a figure, you say the effect is roughly of X size, right? If it's possible to say that, and like you said, I don't want to say stress has an impact, right? That's right, because an impact could be positive or an impact could be negative, right? So first off, layer 1, I think we should always say the direction that the effect is, right? And if possible, we would want to put some ballpark numbers to that, right? So this one is kind of an interesting one. So you might say, even after 8 weeks, when all of the control females the control women were healed, we still have 15% of the caregivers not. And at 6 weeks, half of the control caregivers, or the control women, were completely healed, and only 15% of the because again, notice that that's what makes it impactful to the person listening. And if it was your work, you'd say, look, there's a 3 times effect, right? This dropped this by 60, 70%, right? You would use the numbers to illustrate the size of the effect, right? But I like where you're thinking. I never wanted to say impact. I agree with you. You shouldn't say that. Okay, alright, so I can hand these back. Let's see. Alright, so they're in alphabetical order. So if you're at the beginning of the alphabet with your last name, you should come down first. We can kind of make that a little bit easier. Yeah, I left your extra pages. Yeah, I just forgot. Oh. Yeah, amazing.
It's hard, yeah. I was like, I was going anyway. Oh, I know, but then college happened.
I was like, y'all sure y'all fine. I didn't even look. I don't know what I got. I ain't looking, no. That's incredible. That's incredible. I think I got an A.
Okay, yeah.
I got an A. God is good. God is good. I tried like everything. I got an A. I can't believe it. God is great, bro. That's crazy. God is great.
I'm about to see.
God is great.
How the days go.
God is great. Wow. I followed everything I had. I followed everything.
My name's not that
Oh
far.
my God.
I'm proud of you.
Thank you, man.
Yeah.
I fought for that.
I mean, you brought the little
Oh yeah.
duck and Darth Vader. I said, you gotta do well now.
Man, I fought for that. It was all night studying that day. I came straight in. Yeah. Man. I needed that, Lord. Thank you. Oh man. I thought I had got a 50 or something. I thought I got a 50.
You need to have better faith in
I
yourself.
went
You
to
did
his
an
office
all-nighter.
hours and said that. I was like, what happened if I got a 50? He's like, just wait and see.
How'd you do on the race ride?
Man, that was incredible. That's incredible.
Sorry.
Now you gotta do good on the next one.
Yes. Thank you.
Hopefully kind of an all-nighter.
Yeah, right.
Yeah.
Gotta figure it out.
Earlier this time too.
Same. I got to. I needed that. Thank you, Lord.
I sent it to you. To me? Yeah. I didn't get it. I sent it to you today.
That's what I was talking about. That's why I was like, are you okay? Like, when did I not respond? That's what I was talking about.
I came here, but I was like, I think it's good.
Okay,
I'll keep sending it though.
Yeah.
I think it's.
Oh, yeah, this is the one I was saying. I just heard it. If you're talking about, I think I got. We mixed it up in New York City too. When I was writing down, I was like, wow,
Brought it over. Okay.
I got some Marshalls.
All right,
Parents
so
ratifying.
quickly, if we can, so generally, I want to make a few comments. I think the exams are getting much better. I mean, now, this one was a little bit longer. I put a few extra questions on here, so I think the total possible is like 120 possible points. So to kind of help, right, with the averages from the first grade, there was many more points, so that there won't be as many points on the next exam because it's also longer for you to take the exam, but, you know, anyway. All right, so appetitive and consumatory were the two, right? Appetitive goes with estradiol, consumatory goes with DHT. So, already okay. Estrogen, E2, estradiol, right? Any of those were acceptable. VMN is the brain region. California mouse males. Testosterone was the one that I was looking for, but if you put prolactin, you know, I gave it to you because it is true that they go up, even though testosterone was what I was sort of after there. That's okay. Prolactin increases broodiness in birds. Progesterone is the first one, and then serotonin is the neurotransmitter. And the last one, there were a lot of answers to this, but there had to be some mention of some physical touching of the flanks, right? Because it is a reflex. Yeah?
Can we go over the blanks for 4 and 5 again, please?
Prolactin.
Uh-huh.
And 5 is progesterone. The first one is progesterone. And the second one is serotonin.
Thank you.
Yep. Okay. So, I'm going to make the longest point about number 1 and 2 for the short answer. All right? People struggled with these significantly. So, I want to. So remember, when the number one rule when designing an experiment, whether it's in real life or theoretical, is keep it as simple as possible. Right? That's job 1. So if you are measuring motivation of any type at all, what kind of test will you use?
Obstruction.
Some kind of obstruction. It could be a bar, it could be a wheel, it could be a wall, it could be an electrified grid. It really is inconsequential. So really, you can do this basic experiment in kind of one of 2 ways. You can have a control group and a group that gets the drug. Both of them get the same obstruction test and you compare, or you could have an individual serve as its own control, right? So they take the obstruction test under one condition and then they get the drug under the second condition, right? Because again, the question is, does this affect drive? So a lot of students try to map it onto the like high, low, medium, there's like lots of castrated rats and guinea pigs and all of that. Should they be intact? Can they be intact? And we answer our question.
Yeah.
Yes, then they should be intact. Right? Because what we're interested in, and later on in the exam, we learn, hopefully, and most of